So the next thing, after you have collected the sample, remove the tissue that you need. The next thing you have to do, you have to document the detail of the sample. So this is very important. So you have to label your sample properly. So usually the proper label, you will have a voucher reference number. Okay. You will have a location information, who collected the specimen, when the specimen was collected, and also taxonomy information and the photo. So all this information is very important when you preserve and collect the sample. Okay. So after you have collected the sample, okay, then you have to keep the sample properly. Okay, usually you will keep in the collection or museum. So you have the voucher specimen. So this can be the leftover of the organism. So it's just for the organism, you just take part of the tissue. Then the remaining of the organism, you just keep it and put it a proper label. And after that, you have the physical specimen. You also need the specimen information. So specimen information is the information that I showed just now regarding to the taxonomy, location, collection date, and others. After you have these two, then the next thing you can do is to proceed to the molecular work to get the DNA sequence. Okay. After you complete your study, then all this information need to be integrated and cross-refer to each other. So we're going to discuss more of this during the DNA barcoding and also the sequence repository database. So we're going to discuss this in the future in more detail. So you just have to remember you have to collect all the data and keep it properly. So after that, okay, after you take a piece of tissue from an animal, okay, and then you already label them properly. So the next thing you can do is to preserve the tissue. You have to put the tissue in chemical or in a fridge to preserve the tissue and also the DNA.